The objective of this method is the detection and quantification of double-stranded DNA using the reduction of tetrazolium salt to form a visible precipitate.
The DNA staining protocol has 3 general steps. The first is to put the gel in a solution of Cybergreen 1 and nitro blue tetrazolium, also known as NBT. The gel is then stirred for 25 minutes if it is a polyacrylamide gel and an hour if it is an agarose gel. The last step is to expose the gel to blue light source such as blue LEDs or sunlight. During this step, some precipitate bands appear. These bands have a direct correlation with the DNA concentration, and the gel then can be digitalized in an office scanner in order to quantify the DNA. For the polyacrylamide gel, a non-denaturing polyacrylamide gel is prepared. The gel is then loaded with the samples and the electrophoresis process starts. Cybergreen 1 diluted to 1.2x and the NBT 0.20 millimolar solutions are now prepared. The gel is put into a plastic container